Hello. Hello, everyone. Am I audible? Okay, sir. Okay, sure. So we'll wait for another 2 minutes till then everybody is joining up. Then after we'll start the session. And as we can see, our invited talk and the session chair is also available. We'll start the session at 11:05 sharp. Okay, sir. Okay, sure. Hello, everyone. So as we can see, our session chair is also available now. We are heading towards the start of the session. Okay, so very good morning to all of you. This is Dr. Viren Pandey from AKS University, Satna. We are at the International Conference on Research, Innovations, and Women Empowerment in Sustainable Agricultural Development. This particular international conference is available or organized by the departmental efforts of Department of Biotechnology from Faculty of Life Science and Technology, AKS University, Satna, with Edu-Mentors Society for Sustainable Agriculture and Rural Development. So from there, we have Mr. Ashutosh Behra. He is available here as a CEO and founder of the society. And in today's session, we have invited talk of Dr. Vishnu Omar. who will be speaking on the topic biological control and biopesticides with the eco-friendly tool for sustainable agriculture. This session will be chaired by Dr. Monica Soni from AKS University, Department of Biotechnology. So we'll quickly look out for their profiles. To start with, we'll start with the session chair, Dr. Monica Soni, who is currently working as an assistant professor in Department of Biotechnology, AKS University, Satna. She is having experience in the field of biotechnology with strong background in microbiology, particularly in the microbiology and molecular biology. She has differential experience in plant growth promoting hormonal work and rhizobacterium efforts as well. In the role, she has also contributed to the sustainable agriculture and focusing on enhancing the plant growth and stress tolerance conditions. She has also worked up in the development and applications of biologically synthesized nanoparticles, and she was available in differential environmental management and biotechnological research throughout her journey. So this is the brief profile of session chair, Dr. Monica Soni.
Thank you, Dr. Viren, for the kind introduction.
Thank you, ma'am. So we'll be introducing our invited talk, that is guest speaker, Dr. Vishnu Umar. So Vishnu Umar, a young professor that is from Department of Entomology, Faculty of Agriculture, Science and Technology from AKS University, Satna. He has been particularly working in the active stages of teachings and research combinedly in entomological and sustainable pest management. He is having a vast experience, but in the past 3 years, he has contributed more on sustainable and eco-friendly methods for insect pest management, and he has focused on the approaches to meet up the environmental safety. His work has contributed in the development of biological control strategies and integrated pest management, that is IPM, practices, which is becoming the modern day agricultural demand. Today, he will be sharing us detailed insights on the topic, biological control and biopesticides, and eco-friendly approach for sustainable agriculture. Before starting the session, I would like to welcome Dr. Vishnu Umar, and his experience will be delivered here. And I guess the participants will be benefited from the particular invited talk. Dr. Vishnu, good morning and welcome in this session.
Good morning to all. Thank you, sir, for your kind introduction. So without taking much of valuable time, I would like to begin my presentation on topic biological control and biopesticide and eco-friendly approach for sustainable agriculture. So
okay,
first one is share.
Dr. Vishnu, sir, sorry for interrupting. I would just like to share a suggestion that whatsoever questions are available with the participants, they can share after the session so that the talk is not being hampered in between. Okay. Right, sir? Okay, sir. Over to Dr. Vishnu Umar.
Thank you, sir. Now, slide is visible.
Yes, sir, it is comfortably visible.
Okay, thank you, sir. Now, my topic is biological control and biopesticide and eco-friendly approaches for sustainable agriculture. Students, now imagine a world where the food we eat to stay healthy solely harms our health because of the chemical residue present on this food during its production. So nowadays, agriculture not only challenges to produce enough food for our increasingly world population, but also ensure food which should be safe, nutritionally safe, and should be environmentally sustainable. This is where now biological control and biopesticide emerges as promising eco-friendly solution for sustainable agriculture. First one, introduction. Agriculture, as you know that agriculture is the backbone for our food security and rural livelihoods for many developed countries. So that crop production is significantly affected by many insect pests, diseases and weeds. Around 40% of global production due to these happened. To manage this, all we know that farmers only adopted one option, that is chemical pesticide. He relied on traditionally on these pesticides. Although it has chemical pesticide provide quick control, but their excessive and indiscriminate use has created serious environmental ecological problems and many types of residues on our food. These are some problems that created by chemical pesticides. First one is develop resistancy on insects. After developed resistancy, no more insecticide should effective on the population of that. So we should avoid this use of chemical pesticides. Second one is pesticide residues which contaminate our soil, water, and food products, which may affect humans and also animal health, and also uh, problem creating for our beneficial organisms which present in soil also. Third one is chemical pesticides which kill beneficially, such as I tell you, such as pollinators, predators, and parasitoid, so that there is an urgent need to develop eco-friendly pest management approach as adopting sustainable agriculture. Now, sustainable agriculture. What is sustainable agriculture? Students, it is nothing. It refers to the various farming practices which present that refers to the farming practices which meet present food needs without harming environmental and ecological balances or reducing the ability of future generation to produce food, so that its aim to maintain agriculture productivity while protecting environmental health and natural resources. These are some objectives of sustainable agriculture. First one is conservation of biodiversity. Second one, protection of soil and water resources. Reduction of chemicals, pesticides, chemicals, herbicides, chemical fungicides, and fourth, long-term ecological balances. So, for maintaining of these objectives, in this context, eco-friendly pest management strategies plays a vital role. Now, eco-friendly approaches, as you know, there are the various eco-friendly approaches which by using of that we manage insect populations without harming the environment or ecological balances or biodiversity. First one is cultural control, mechanical control, biological control, and using of biopesticides. Among these approaches, biological control and biopesticides have gained significant attention in recent years because of cultural control and mechanical control have been employed by various progressive farmers in the field, and they have not knowledge about the biological control and biopesticide and their effectiveness. Now it's biological control. What is biological control? Biological control is using of or utilization of biological agents such as predators, parasitoids, or pathogens to control increasingly insect population. These are some predators such as ladybird beetles, lacewings, dragonflies, many spiders which directly feed on the insect, which directly affect our crop production, feed on the crop. These are the predators which directly feed on those insects. These are some predators such as Trichogramma, Campoletis chloridi, Isotema javensis,
Dr. Vishnu sir.
Platygaster orygi, and there are
Vishnu sir.
various parasitoids which effectively control the insect population. Now, last one is pathogens, including bacteria, viruses, various
Vishnu sir.
fungi that infect and kill our harmful insect pest. So that these biological control help to regulate insect population naturally. These are some photos of predators, praying mantids, dragonflies, bumblebees, ladybirds, green lacewings, and cactus moth. These are some parasitoids which kill the larva by infecting him. First one is inoculate egg on the insect host. Presentation. Now,
Now it is visible, sir.
okay.
Can you make it full screen?
Sorry.
Can you make it full screen?
Okay.
Sure, sir.
Now, as I mentioned, that these are the some biological controls such as predators, parasitoids, and pathogens. These are the some photos of predators which directly feed on the insect. First one is praying mantids, dragonflies, bumblebees, ladybird, green lacewings, and cactus moth. And this is the parasitoid, such as trichogramma, which directly kill the host by inserting their eggs on the host, and after the hatching of the eggs in the host, they become as larvae and feed on the host and ultimately kill the entire host and uh, complete their life cycle. And third one is, these are the first photo is show that this is the fungus which infect the insects as aphids, and second one is infected by bacteria such as the Bacillus thuringiensis, and third one is infected by viruses such as NPV, nuclear polyhedrosis viruses. Now, biopesticides. Biopesticides are the pesticides which are derived from the naturally, such as botanicals or from the animals. Animals biopesticides such as an eristoxin, and which biopesticide which is derived from the botanicals such as neem, protinol, which is the fish poison also. So that they are derived from natural resources such as microorganisms, also plants and certain biological substances. It is target specific, biodegradable, and safe for humans and also environment. Because of these advantages, they are increasingly popular for sustainable agriculture. There are some types of botanical pesticides, biopesticides. First one is botanical, as I mentioned, such as neem or rotinol. Second one, botanical pesticide, such as microbial pesticide, such as Beauveria bassiana, fungus, and PV, which insect-feeding nematodes. And last one, biochemical pesticide, such as insect pheromones, such as alarm pheromones, sex pheromones. Also, they interfere the insect behavior, which ultimately kill the insect. There are some advantages of biological control to promote sustainable agriculture. It is environmentally safe, as it is key that biological control में जो predators हैं या parasitoids हैं, वो they directly feed on the insect and directly managing insect population without pollute soil or water. They are target specific. They are not affecting the beneficial organisms. They maintain ecological balances and provide long-term pest suppression. And there are some advantages of biopesticides also. They are biodegradable and leave very little residues on the crops. Second one is, it is relatively safe for humans, animals, and beneficial insects. Third one is, they are cheaper and some of can be made by own farm inputs by farmers, such as neem, brahmastra, neemast, and there are various biopesticides which have applied by the farmers on the farms to kill insect population or disease problem. And their role in sustainable agriculture, as I mentioned, they help reduce dependency on chemical pesticides. They support biodiversity conservation and protect natural ecosystem. And promoting ecological balances, these approaches enhance the sustainability of agriculture production system. And last is conclusion. Excessive dependency on chemical pesticides not sustainable in long-term. Students, biological control and biopesticide provide environmentally friendly and effective alternative for pest management, and their wider adaptation can contribute significantly to sustainable agriculture, environment protection, and food security. Thank you all.
Very well delivered by Dr. Vishnu. Now the session is open for questionnaire. So participants can share your questions directly to Dr. Vishnu and he will be answering them. Anyone having any query? So Dr. Vishnu will wait for another 1 minute. Then after the
Okay.
session share, she will be having an one-to-one interaction with you.
Okay.
So I guess the participants are having no queries. Over to Dr. Monica Soni, session chair, for one-to-one interaction with Dr. Vishnu Omar. Monica ma'am, am I audible? Yeah.
Yes, Dr. Viren, you are audible. Yeah. Who is the first presenter here?
Dr. Vishnu.
No, after Vishnu, sir.
Okay, so for presentation,
Yeah. Sir, I have a small questions regarding your work. Sir, have you prepared any biopesticides?
Yes, ma'am. In AKS form, we have prepared such as Brahmastra and Nimastra and
Okay.
Suthast and some of the biopesticides which have been prepared by the Department of Entomology on AKS form.
Okay. Thank you, sir.
Okay.
So, shall we start, Dr. Viren, for presentation?
yes ma'am, we can do that.
Yeah.
So let me check who is available for the first session. Any participant who would like to take up the opportunity as early bird?
Shall I go first? I am second only, so I'll go. I'm Pranita.
Sure ma'am, sure ma'am.
Okay, so let me share my screen. I am doing it in mobile, so let's see.
No issues ma'am, we'll help you out.
I'm sharing your screen. Can you see?
Ma'am,
Yeah,
it is-
it's visible, but you can enhance the slide, please.
What?
When there is a play button in the below portion, you can click it.
Okay. A play button. Okay.
Yeah.
So
Now-
is it visible
Now it
now?
is available. Yeah.
Yeah. Thank you so much. So I'll be talking about how we can use transcriptomics to deal with the drought stress in plants, right? So just a small introduction about the problem. So drought stress is now a very challenging for us, like considering the climate change and some areas where the rainfall and all they are less, and we are having water scarcity. So there are some plants which are very much drought tolerant, stress tolerance. Among stress, we can talk about salinity, drought, heat, cold, many things. But today I'll be dealing only with drought stress tolerance. So it is one of the major abiotic stress that is reducing the global productivity that we are having. And since we are an agriculture-based country, so climate change is going to affect us a lot. So now what scientists are doing, they are trying to understand the molecular mechanisms by which some plants show tolerance towards drought. So basically, what are the strategies that plants adopt to deal with plant stress? So there are three types of strategies. The strategies like first is drought escape. What they do is they complete their life cycle in favorable conditions only, and when the drought comes, they are done with their life cycle. So that is one way to survive. Next is to avoid the drought, like the tomato closures and to reduce the leaf area. The xerophytic plants, whatever the xerophytic plants have mechanisms, deep root system and all. So they avoid the drought by using these mechanisms. And the third is to tolerate the drought by changing their metabolic processes or adapting to these processes during water deficient. So these are the processes which plants adopt. So the physiological and biochemical defense mechanisms that plants have, they adjust the osmotic things like with some amino acids like proline, sugars, and glycine, betaine. They adjust the osmotic pressure and all. Another mechanism is antioxidant, like during stress, antioxidant reactive oxygen species are created, which disturb the other metabolic processes of the plants. So by the activation of enzymes that absorb those species or change it into less harmful things, so that is like antioxidant defense mechanisms are adopted by these enzymes. So SOD and peroxidase, these are some enzymes which have been seen to manage these kind of responses. Another is hormone regulation. By regulating the hormones, like we all know, the abscisic acid is is helpful during the stress. So by upregulating the production of this hormone, also drought stress can be tackled. Next is some metabolic changes, creating like shifts in the photosynthesis mechanism and energy metabolism. By changing energy metabolism, this is also one method by which plants deal with stress. So this in this diagram you can see these mechanisms what the plants adapt to tolerate stress. Okay, so now transcriptomics involves to like study all the genes that are expressed during the stress, drought stress. So if we know the mechanism, the genes that are responsible for managing drought, we can use these information to in breeding experiments or in or in adapting the same mechanisms in other sensitive plants. Okay, so that is basically transcriptomics. So for those who do not know what transcriptomics is, like we study the expression of the genes, the transcriptome we see. Transcriptome means just like genome, we say genome is like all the genes present in the cell. When we talk about transcriptome, it is talking about all the genes that are expressed during a certain condition or during or in a certain tissue. So that is transcriptomics, the whole of messenger RNA that is expressed in a cell. So the complete set of RNA that the plant or any organism, any cell is expressing at a particular time. So there are methods, RNA sequencing, microarrays, next generation sequencing has been very helpful in studying transcriptomics and RNA sequencing. So what we do is we take a control and we take a drought prone plant to which we are giving drought stress, and then we study the differentially expressed genes, like what genes are expressed in a drought plant and but not expressed in the normal one. So we can say that okay, these are the genes which are responsible for managing drought. So some of the genes are like heat shock proteins, aquaporins, leaf proteins. They have been known to help in drought stress management. So the methodology is here. If you want to do it, we prepare the sample, we expose the plants to drought conditions, we keep one control, and then a library is constructed. The whole RNA is extracted and it is converted into cDNA, because working with RNA is a little difficult. So we convert it into cDNA and then we sequence it. After sequencing, we align it with the genome to understand what are the differential genes that are expressed in the drought the plant which is exposed to drought conditions. So we align it with the whole genome. If the whole genome is not available, then maybe we can go for like another alignments with closely related plants. If that genome is available, then we can go for that also. Then we map to understand the pathways. We use gene ontology and KEGG's databases to understand the pathways that are involved. So I will be discussing two experiments, two researches that have been done relating to drought tolerance mechanisms. So actually there are many in many plants like rice, wheat, and many pulses also. This work has been done, and still a lot of work has to be done. But I'll be discussing in two plants. So in oats, what they did, they took two varieties, a drought tolerant variety and a drought sensitive variety, and they did RNA sequencing or transcriptomic analysis for both the plants. And what they found was that the antioxidant enzymes, the expression of antioxidant enzymes were increased, like SOD and CAT. These are enzymes. And they found that leaf wax was increased, stomatal structure was changed so that it retains more water. And then some regulatory pathways were discovered, many differential express genes involving cell wall and wax biosynthesis. They were also found. And these are some of the findings which are common to many plants, like the work done on rice, wheat. So these are some of the common findings which they have gotten in many drought tolerant plants. So and also there are some transcription factors, transcription families. So what transcription factors do? They manage the expression of these genes. They are not themselves involved, but they are involved in the expression of the genes that are directly involved in stress management, drought stress management. So these are some of the transcription factor families, Warki, NAC, and these have been found in many plants. MYB, YB, BZIP and all, abscisic acid response, they manage and regulate abscisic acid response. And so also these transcription factor families have been commonly found in drought tolerant plants. Then coming to long non-coding RNAs. So now a lot of work is going on long non-coding RNAs. Earlier it was felt that those genes or those RNAs which are not coding for any protein, they are useless. They are not useful. But now a lot of research has found that even the RNAs that are not making proteins, they are also important in the metabolic functions, because what they do is that they regulate the expression of other genes. So many long non-coding RNAs, they are hidden regulators. Why we call them hidden regulators? Because they are not themselves being expressed, but they are controlling the expression of some genes like LEA3 and VOC genes. And so the plants that are overexpressing these genes, they have been found that they are having large number of these long non-coding RNAs. And the research on long non-coding RNAs is very primary recently, and a lot of work is still needs to be done. And like this is a very like if we talk about future prospects, then we can work on long non-coding RNAs in the major drought tolerant plants such as millets and all. The millets are very much drought tolerant. So these are some of the metabolic pathways which are affected by long non-coding RNAs. So photosynthetic efficiency, the long non-coding RNAs help in increasing the efficiency by maintaining the chlorophyll, because drought and these reactive oxygen species can damage the chlorophyll structure and all. So they help in maintaining the structure and its metabolism during stress. Then lipid management, since we all know lipids are
Dr.
anybody?
Pvt,
Yeah.
Ma'am, we have only 1 minute to wind up.
Okay. It's going to be over. Yeah. So cell membrane lipids are the members of cell membrane. So if the cell membranes and cell walls are intact, then drought water management will be done efficiently. And then detoxification, again, the reactive oxygen species and compounds like methyl glyoxyl and all, they needs to be removed. So they are also involved in doing all this. So in conclusion, I will say that drought management is an integrated response where not only one mechanism is involved, but many mechanisms like roots, cell wall synthesis, photosynthetic efficiency, wax deposition, and managing the reactive oxygen species. All the pathways, they play an important role. So if we get the molecular markers that are responsible in managing drought, then we can transfer these. They can be helpful in selecting varieties for breeders, and also they can be used to transfer it in other crops for drought management. And these are some of the future prospects which I already discussed. So this multi-omics and transcriptomics and all, they are really now very much helpful in managing stress, and not only drought stress, even salinity and other stress. Lot of work has been done, and lot of work has to be done to manage all this. So thank you everyone for your patience. If you have any questions, you are welcome.
Thank you, ma'am. So the session is open for 30 seconds for any query or discussion.
Dr. Pranita,
Yes, ma'am.
thank you so much for your informative presentations, and
Yeah.
you will be presented here. So firstly, I have one questions regarding your work related. That is, have you find out any a differential express gene in drought stress
Yes, actually,
that regulate the osmotic balance and antioxidant systems?
Actually, my work is going on. I am working on guar, that is the guar phali that we all know. It is also a semi-arid crop, and it is quite drought tolerant. Tolerant. So my work is going on. I have not differentially expressed genes. I have not reached that stage as yet. I have done the transcriptomic analysis, and now I am doing this transcriptomic studies analysis that is going on. So I am expecting to find some new nice genes, and maybe the common genes will be there already. So when I'll be done with this work, I may share with you. Yeah.
Okay.
Okay. Thank you, Dr. Pranita. Now over to Mr. Anurag Tripathi. He will be presenting his talk, and we have 8:2 as a timing. So make sure the participants are visible, not visible with the timing. Sure.
Okay, thank you.
Yeah, Mr. Ashutosh.
My speaking is good.
Anurag, can you please hold up for 1 minute?
Yeah, sure, sir.
Yeah, Dr. Pranita, Mr. Ashutosh is having one-to-one interaction with you regarding your presentation. Over to
Yeah.
Ashutosh, sir.
Yes. Yes. See what it can be helpful in, like in drought tolerant, and we can use them in breeding experiments. We can cross them with some variety, with the high yielding varieties. So the high yielding varieties will also get will be drought tolerant. Plus what we can do is that the two in by like considering the next level, we can transfer these genes in drought sensitive plants. If that is successful, then that it will be useful that way. Hello. Yeah.
Okay. Thank you, Mr. Ashutosh and Dr. Pranita. So if permission is available, we can start with Mr. Anurag Tripathi. He is available with his presentation.
Okay. Thank you, sir.
Okay, over to Mr. Anurag.
Okay. Thank you, sir. Good morning to one and all present here. My name is Anurag Tripathi, PhD research scholar in the Department of Agriculture and Meteorology from Pandnagar University. My topic of presentation is seasonal variation in thermal indices and heat use efficiency of maize. Here is a brief introduction about the maize crop. As we all know, it is a queen of cereals, and because of advantage of adaptability and high potential, we are growing in a larger way. And we know that it is grown in more than 160 countries and a major source for food, feed, and fodder for all the human beings. India, maize is the third most important cereal crop after rice and wheat and play a crucial role in food security, poultry feed, and agriculture industry. In the right side, you see the experimental I have done. So I start with the treatment itself. I use the split plot design in which two factors are there, main factor and sub factor. Main factor, I choose the three different varieties. One is composite and two are the hybrid varieties. And in sub factor, I use the different fertilizer doses. One is the control, second is the RDF 80% plus vermic 20% on nitrogen basis. Third one is the RDF 80% plus 40 manure 20% on nitrogen basis. And the fourth one is the RDF, that is the recommended dose of fertilizer. Also, I have done a two trial, winter and spring both season in 2024-25, and in the right side you see the treatment combination. So I have calculated the thermal base indices using the field trial. What in which I have calculated the GD, that is growing degree day. It tells about the linear relationship between the crop growth and the yield. It is calculated using the formula of T maximum plus T minimum divided by minus T base temperature. Here T maximum and T minimum are the daily maximum temperature of the particular location, and taking the average of that and minus with the T base temperature. T base temperature is depend on the genotype. So for the maize crop, the T base temperature is 8 degrees centigrade. By using this, we can calculate GAD. After that, we calculate photothermal units. It depends on the day length of a particular day. So by using the formula GAD multiplied by day length, we calculate the photothermal unit. And third one is the heliothermal unit. It is calculated on the basis of actual bright sunshine hour of that particular day. So here the formula is multiplying GAD with the actual bright sunshine hour to get the heliothermal units. I'll see the results. Now this is the number of days taken to attain different phenophases in winter maize. Here you can see the treatment in the horizontal direction, and in vertical direction we see different phenophases of the maize crop. So here it says that D1 T1 takes 13 days from sowing to emergence. Like that, we'll see at maturity the minimum days taken in the winter maize is the 132, and the highest days taken at B3 T4, that is 151 days from sowing to the harvest at maturity. And for the spring season, we'll see the maturity is attained at 92 days for the lower and the 110 days takes at the higher days. Now, this is a heat map of GAD accumulation across treatment and phase in winter maize. GAD, as I said, it is calculated by taking the average of maximum minimum temperature on daily basis by minus with the T-base temperature. And here I have calculated at the here we'll see maturity right in the right corner. We'll see the maturity. The dark value shows that it takes the higher GAD. The higher GAD shows that it gives the maximum yield. This is a concept. It shows a linear relation between temperature and yield. So V2, there is 12 treatment, that is V3 T4 shows the maximum value is 1472.2, and the minimum value is attained at treatment, that is V2 T1 is 1146.1. Same trend is followed in the spring maize. The 12th treatment giving the maximum GAD, that is 1848.7, but the minimum is observed at first treatment, that is 1500. In the same way, I have calculated the heliothermal unit. It is based on the actual bright sunshine hour. So here 12th treatment giving the highest value at maturity, that is 11,363, and the minimum value is observed at the 5th treatment, that is 8,319. And for the spring maize, I have again performed same pattern and observed the same pattern also. In 12th treatment, it gets the 15,858.3, and the minimum one is observed at the first treatment, that is 12,851 maturity. Now the photothermal unit, it tells about the how efficiently the physiological maturity is attained depend on the day length of the particular day. So for the winter maize, the maximum PTU observed at the maturity stage at 12th treatment, that is 16,894.5, and the minimum observed again at the fifth treatment, that is 1,748.6. And for the spring maize, same trend is followed. At the maximum value is obtained at 24,554.4 at maturity, that's the 12th treatment. Now we use the thermal use efficiency. It's about how efficiently the crop uses the thermal in growing season. Here in the left side we have the winter maize, and in right side we have the spring maize. Here we'll see HUE is there, that is heat use efficiency, HTUE, that is heliothermal use efficiency, and photothermal use efficiency. Here V1, V2, V3 are the three varieties. In the varieties, V3 perform well by the thermal unit and gives them highest yield. In comparison to winter and spring, the spring one is perform better as compared to the winter one. And in fertilizer basis, we can see that the F4 treatment perform well in both the scenarios. But in comparison with winter and spring, winter one is giving the best result for the fertilizer consumption. We'll see it by using a graph. Here, this graph shows that the left one is showing the variety difference in both the seasons. The variety performance, winter, the thermal use efficiency of winter is slightly lower than the spring one. So using the same variety in the spring season gives the best result. And for the fertilizer basis, that is in the right side, we'll see that the same fertilizer doses is given in the spring and winter season. But in winter season, the use efficiency of the fertilizer is very good as compared to the spring. Now, this is the relation between the yield and thermal indices of winter and spring maize, as ultimately we are doing trials for the yield. So here I'm comparing all the thermal indices with the yield, and we'll see it by using regression line. Here we have seen that the upper one is for the winter, and the lower one graphs are for the spring one. The upper one shows for the GDT and HTU and PTU. The R square value is around 64%, 0.64. So around 64% gives a best result. And for the spring, the values are around 70%. It varies for GDT. It is around 0.70. For HTU is 0.72, and for PTU it is around 0.70. So by this we can see that the spring one is giving the best use efficiency with the GDT, HTU and PTU in the spring season as compared to the winter one. Conclusion: the study showed that the thermal access significantly influenced the growth and yield of spring maize. Spring maize accumulated higher thermal units and GD ranging from 1500 to 1848 degree days compared to the winter one, that is the maximum it is at 1472, resulting in better crop performance. Among the varieties, V3, that is DKC 9188, is the Bayer variety, recorded highest heat use efficiency in the winter and spring season. And on fertilizer basis, F4 is 100% recommended use of fertilizer, produce the highest thermal use efficiency and grain yield with the maximum yield, that is 12,476.67 kg per hectare, obtained V3 F4 treatment in spring maize. So therefore, V3 variety with 100% IF was found suitable for this particular location. Thank you.
Thank you, Mr. Anurag. Session share, any query with Mr. Anurag?
Hello, Mr. Anurag. Thank
Yes,
you
ma'am.
so much for your interesting presentation.
Thank you,
And
ma'am.
I have one question regarding your work. At which stage you find the maximum GDD accumulation?
It's basically depend upon the different time taken from sowing to maturity. So, sir, the maximum GD is maximum GD, HTU, PTU is observed at maturity stage, because that is the time period which takes maximum duration in the crop. Like from sowing, it gets get at 10 days, 12 days, but at maturity it takes 50 days. So the daily temperature of 150 days gives the maximum GD.
Okay, thank you. And can you apply this method in any crops, in any other crops?
Yes, ma'am. It depends on the different trials. I'm doing work on my maize, so I'm comparing seasonal in winter and spring season. So on thermal indices, pattern is followed.
Okay, thank you.
Okay, thank you, ma'am.
Okay, Anurag sir. Over to the next participant, that is Prince Chaurasia. Is he available? Prince?
I think it's not here.
Okay. So ma'am, we'll be heading towards the next participant, that is Bunty.
Mm.
Bunty, are you available? Yeah, he is available, I guess. Yeah. So you can start your presentation.
1 minute.
Make sure we'll follow 8+2 timing. So you have 8 minutes for the presentation and 2 minutes for question and answer. Okay, yeah, your screen is visible.
Okay, sir. Good morning, sir. And good morning. My topic is system productivity and economic efficiency of maize as influenced by established method and cropping intensity in mid-hills ecosystem. This slide is the introduction to conclusion. Introduction. Global pride and skill of millions of 10 productivity is about 33.62 metric ton and 3.34 ton hectare respectively, according to Government of India. Tillage is a fundamental of agrotechnology operation carried to prepare soil to achieve proper crop growth and yield with minimize degradation, erosion, and greenhouse gas emission through practical like minimum conventional raised plantation, adoption of an intensification cropping increase system productivity and equivalent yield by 25 to 30%, improve resource use efficiency also, higher economy returns. So objective mainly to study the effect of fertilizer method and cropping intensity on system productivity and use efficiency of maize crop. To work out the relative economics material method, use split flow designs and varieties use VL45 and gross load size 5.4 into 7 or 4.70 meter, and spacing is mid spacing is 60 centimeter into 30 centimeter, and so intercropping showing in the middle of main crops. Location my experimental location is a research farm RARS Rajouri Square Jammu. In these are the soil study status of experimental field. Soil texture is silty clay and OC is medium. Available nitrogen is low and available phosphorus and available potassium is mediums. Treatment details in main plot slab method is a conventional tillage, minimum tillage and raised bed, and subplot is a soil maize plus maranthus, maize plus cowbee, maize plus French bean. Intercropping ratio 1:1. Result effect of tillage method and intercropping of plant height of maize in the tillage method is showing higher and significantly showing in treatment 1 and statically at par in treatment 1 and low and minimum in treatment 2. In intercropping is maximum plant height is at C1 and statically at par is C3 and C4 and minimum is C2. And effect of the tillage method and intercropping of maize dry matriculation is in tillage method is significantly higher in treatment number 3, and treatment 1 is statically at par, and maximum in treatment 2. Intercropping is highest in C1 and statically at par in C3 and C4, and minimum in C2. And effect of tillage method of intercropping yield of maize in this also same trends, plant and dry matter, but in effect of tillage method and intercropping of maize equivalent yield and stem productivity is highest in the maize productivity, and maize equivalent yield in treatment 3, and growing by the treatment 2, and minimum in treatment 1, and intercropping is was in maximum iron treatment number C3, and statically at partner C4 and lowest in C1. Conclusion, it may be concluded that under mid-disease condition of raspberry growing, maize growing of raised bed according to higher crop growth, plant height, and dry mat regulation, seed and destroyed and productive as equivalent yields, whereas C1 is maize system productivity, higher growth plant height and dry mat regulation, seed and straw yield, but study of the was found in the treatment C3 maize plus coffee. Thank you.
Thank you, Mr. Bunty. Dr. Monica, any query with Mr. Bunty?
Hello, Mr. Bunty. Thank you so much
Yes,
for your
ma'am.
presentation. So my question here is, have you established any method for the cropping intensity?
Yes, ma'am.
So what method?
मैम जो मेरा ट्रायल है मैम ईयरली है मैम, ये राजौरी रीजन के लिए वहाँ पे मैम अभी मेरा मस्टर्ड के साथ चला हुआ मैम, मस्टर्ड के साथ भी मस्टर्ड प्लस में वीट है, मस्टर्ड प्लस में गार्लिक और मस्टर्ड प्लस में फील्ड पी है। तो ये
अच्छा।
मेरा मैम ईयरली है, क्योंकि हमारा मेन मोटिव है मैम राजौरी में कोई भी काम अभी तक हुआ नहीं है, तो वहाँ पे एक पैकेज ऑफ प्रैक्टिस बनाना है ये रेन फीड एरिया में राजौरी इली एरिया में, तो इसलिए हमने पूरे ईयर के लिए टिलेज प्रैक्टिस और ये क्रॉपिंग सिस्टम लिया है।
Okay. Okay. Thank you, Bunty.
Okay. Thank you, sir. So we'll be moving to the next participant, that is Sanjay Kumar. Is he available? Mr. Sanjay, are you available? No? Shall we move forward? Okay. So we'll be heading to Shivani. Shivani, ma'am, are you available? Yeah.
Yes, sir. Yes, sir.
Okay. So please share your PPT.
Okay. Is it visible, sir?
Yeah, ma'am, it is visible.
Okay. A very good afternoon to esteemed scientists. I am Shibani, a PhD scholar in Division of Agriculture Extension Education at SCOS Jammu. Today, I am presenting my presentation on livelihood diversification among farmers practicing integrated agri-aquaculture farming system in subtropical Jammu region. So let's start with the introduction. As we all know that the landscape of Indian agriculture has shifted dramatically. In 1950s, 69% of India's workforce was engaged in agriculture, which contributed 53% to the national income. Fast forward to 2025-26, this proportion has dropped to 46% with the share of 18.2% to the total GDP of the country. Our workforce or our farmers in India significantly facing problems like fragmentation of land, diminishing farm size, soil degradation, excessive use of fertilizers and agrochemical, and most importantly, climate change, which causes serious problems to the production, socio-economic and environmental sustainability of agriculture. If we say farmers depending on single enterprise like monocropping system, which is not sufficient to sustain the livelihood of the farmers. So to overcome these problems, integrated agri-aquaculture farming system come in. This is the system where agriculture combined with the fishery and livestock helps to combat with the poverty, hunger, and sustain the income of the farmers. If we talk about specifically about Jammu and Kashmir, a vast amount of wasteland and waterlogged land in farmers field offer immense potential for aquaculture development. Many farmers are recognizing the benefits of integrated aquaculture with traditional agriculture, which leads to the diversification and ultimately increase the productivity. So these were the objectives: to examine the extent of livelihood diversification among farmers practicing integrated agri-aquaculture farming system. And the second one was to identify different livelihood activities adopted by integrated agri-aquaculture farming system practicing farmers. Moving to the material and methods, Jammu and Samba district from Jammu and Kashmir were selected purposely because these districts were come under a subtropical region. And for data collection, 20 farmers were taken, each 10 from each Jammu and Samba district. A well-structured interview schedule were developed to collect the data and to analyze the diversification pattern. Descriptive statistical tools such as frequency, percentage, and mean were used. Now coming to the results and discussion, as you can see that majority of farmers were that were 55% engaging in 3 activities, and 20% of the farmers were engaged in 2 activities. A small portion of farmers, that is 15 and 10%, were engaged in 4 activities and 5 activities. Average diversification of the average diversification level was 3.15 activities. That reflect that farmers rely on the combination of 3 activities to strengthen their livelihood. So if we talk about the different activities they were in which they were engaged, so they were engaged in both on-farm and off-farm activities. In on-farm, they were performing agriculture with fishery, dairy, poultry, and goatry. And in on-farm activities, they were also doing as a labor, and some of were shopkeepers, restaurant owners, drivers, and even ex-servicemen, which collectively contributing to improving their household income and livelihood stability. So in conclusion, I would say that the integrated agri-aquaculture farming system is a proven tool for improving household income and stability. By diversifying into an average of 3 activities, farmers are effectively mitigating the risk in traditional farming. And moving forward, it is important and vital that we support and we extension system support farmers through different extension services and institutional interventions. If we can scale this model, we can significantly strengthen rural livelihood across the regions. Thank you.
Thank you, Ms. Shivani. Ma'am, Dr. Monica, ma'am.
Yes, thank you, Ms. Shivani. Thank you for your presentation. I would like to ask about your research work. What is the sample size you have chosen here?
Ma'am, basically it was pilot study. I
Okay.
have chose 20 farmers, 10 from district Jammu and 10 from Samba.
Okay. And how does the integrated farming system help in improving farmers' livelihood security?
Ma'am, if we depend on a single enterprise, like farmers depend on monocropping, so due to climate change and other risk they facing, the income they generate from a single enterprise is not sufficient for the stability of their livelihood. So if they are integrating in different enterprise or different activities, and if their family size is also large, they can combat with the market risk or financial stability.
Okay, thank you, Ms. Shivani.
Thank you, ma'am.
Thank you, Ms. Shivani. So we'll be moving to Divya Gautam.
Yes, sir. Good afternoon. So, sir, my slide is visible.
Yeah, ma'am, it is visible.
Yeah.
Can you play that?
In a full screen, Divya,
Yes, ma'am.
can you play in a full screen?
इसमें हो नहीं रहा है एक मिनट
So in case Divya, if it is not available, we can continue with the other participants and you can rejoin. Ma'am, is it comfortable? Dr. Monica, is it comfortable?
Yeah, I'm comfortable.
So we can start with the new participant.
Okay.
So she is with the PDF. So Dr. S.K. Mohammed Ashif. Okay, he is not available, I guess. Divya, can you please switch off your mic? Okay. So next we have is Rashi Mongia.
Yes, sir. Morning, sir. Thank you so much for inviting me. Hello, Monica ma'am, and hello to all the co-research fellows, as well as all the eminent scientists. Thank you so much for inviting me, and it's a huge privilege for me to speak in front of learned people like you. I am Rashi Mongia, research scholar of AKS University. I am pursuing my PhD under the guidance of Dr. Ashwini Rao ma'am. Thank you so much for guiding me, ma'am. Shall I proceed?
Yeah, ma'am.
Yeah.
Thank you so
Proceed.
much. Yeah. Just give me a moment, ma'am. Ma'am, is the presentation visible?
No, ma'am.
Yeah,
Yeah,
just-
now it is visible.
Okay.
Yeah, now it is
Thank
visible full screen.
Yeah. Thank you so much, sir. Okay. The topic of my research is heavy metal detection and microalgal treatment of contaminated water. Now, as we are in developing countries as well as in developed countries, there's one major global problem which is coming across, that is heavy metal pollution. Although we are industrializing and we are moving forward in life, but we are ignoring the fact that this industrialization has led to a lot of heavy metal pollution. So let's work on that. And I've tried with my guide to work on a little, inexpensive method by using biological microorganism. So the hero of my research is microalgae, undoubtedly the global player of biological remediation. It is often called as liquid gold because it has the capability to produce everything right from fuel to food. As well as for sustainable remediation, it is one of the key players. And the factor which I'll be working on it, I'm working on the bioremediation factor of microalgae. Heavy metals generally enter into the water system. Now the process through which they enter, first is industrial process, then mining activities, domestic inputs which might enter into the lack of a sewage system or improper sewage system, which may lead to water pollution. Now the study area which I focused on was the areas where wastewater collected through some of the area. So I chalked out few areas and collected water from that. Sampling technique which I used was simple grab sampling technique. Plastic bottles, disposable water bottles were used and samples were collected in it. And as I had to do metal detection, the samples were collected in it and then transferred to private laboratories for AAS method. Now, why are we so concerned about heavy metal? There are so many kinds of pollution, but why am I working on heavy metals? This is the reason behind. Because heavy metal is entering into the biological chain and impacting every factor. And the factors which are seriously impacting the health of human as well as animals and the entire aquatic system. That's the reason why I am working on heavy metal removal. So first is you may cause serious kidney dysfunction. Lead is most probably responsible for nervous disorders. Then you have skin diseases, immune system effect, and many of them, for example, chromium is also carcinogenic. Specific toxic mechanism, they enter into cellular level as well as enter disrupt metabolic disruption, and various other techniques are used by which through which they enter into food chain and disrupt the life of human. Now the objectives which I proposed were, firstly, I wanted to gather the data of heavy metal concentration, and then by gathering it, my next step would be isolation of microalgae from the indigenous site, and then using the same microalgae for the remediation of wastewater. So this is what my objectives were. And then finally, I'm going to treat it with the microalgae. Now, results which I have undergone till now in my ongoing research is, I have taken samples from 2 of the places of Satna, and I have observed, I sent the samples through a laboratory, and the research which the result which I came across, I got concentration of lead and chromium were visible, and they were pleasurable content was found in the water. Now the ongoing research is under the process. Now the future directions, if I would like to suggest is, obviously, as I said, microalgae is the global player. So he's the global player of remediation and the cheapest method, and we are providing a better solution instead of going into expensive chemical or physical methods of purification of water. With that, I would like to rest my presentation. I'll just stop my screen sharing.
Yeah, sure, ma'am. Thank you, Rashi Mungia. Dr. Monica, any query with Rashi?
Thank you, Rashi ma'am, for your informative sessions here. I have questions you. Firstly, that is which method you have used to remove the heavy metals in water?
Ma'am, AAS, atomic absorption spectrophotometry.
Okay. And any microorganism used for the treatment of removal of heavy metals?
Ma'am, I am focusing on microalgae. So I'm trying to work out with microalgae, and the species which generally help out in heavy metal bioremediation are Chlorella vulgaris.
Which species you have used in the removal of
Ma'am,
heavy
I'm not
metals?
used. I'm going to isolate from the indigenous sample. I'm not using it, applying it from the outside. I'm going to isolate from the polluted site itself, so that even that is cost productive, and the microorganism is already adjusted to the environment, and I don't have to apply extra anything to that environment. So I'm going to isolate the indigenous microalgae from the site.
Okay. Okay. Thank you, Rashi.
Okay.
Thank you so much.
Yeah, thank you, Rashi. So
Thank you.
we'll be moving to the next participant, Dr. Muhammad Asif.
Okay, sir. Good morning.
Good morning, sir.
May I share my screen, sir?
Yeah, sure, sir. Asif, sir, can you switch on your camera as well?
Okay, sir.
As we are live,
1 minute, sir.
so participant can see you as well. Now, I guess Muhammad Asif will having concern with IT, I guess, as he is out of participant list. So we'll be moving to the next participant.
Sir, can I share my screen?
Okay, Divya,
Just where you can share.
sure. Till the time Asif sir is joining, you can continue.
Sir, screen is visible or not?
Yeah, it is visible. Ma'am, is it comfortable, Dr. Monica? Is it comfortable for you to just have a look on the PPT?
Not Dr. Viren.
Okay. So Divya, can you share presentation as I can see it is a PDF file.
Yes, sir. I will try.
So the list of participants who are remaining is Muhammad Asif. Next is Humandi Brajwasi.
Yes, sir. My laptop is hanged out.
Okay. So Divya, you work out with the presentation. Till then, we can start with Muhammad Asif sir, as he is ready with his presentation. Over to Muhammad Asif sir.
Yes, sir.
Yeah, your presentation is visible, but you are not visible, sir. Can
Yes,
you
sir,
see the camera as well?
Is it visible, sir?
Yeah, you are visible, sir. Very good afternoon, sir.
Good afternoon, sir. So my topic is phyto-remediations of wastewater using aquatic macrophytes and its valorizations into nutrient-enriched biofertilizer for the climate-smart sustainable agriculture. I am Sheikh Mohammad Asif. I'm assistant professor from the Department of Agriculture of Brainerd University. So currently we know about the scenario of the wastewater pollutions. The wastewater generated through the industry, through the agriculture and other parts are creating a huge problem throughout the worldwide, as the wastewater is rich in nitrogen and phosphorus, and several heavy metals and others organic pollutants. So we have to correct it and remediate it. But the conventional treatment issues like physical remediations or chemical remediations, they require in high operational cost and highly energy required. So this is a problematic. So we can solve this by using phyto-remediations, because here we can grow some plants, and these plants can effectively remove the heavy metals or nutrients and others organic pollutants or contaminant, and that will be low cost and environmental friendly. And we can also utilize the solar driven processes for the remediations of the wastewater. So there are several type of aquatic macrophytes like water hyacinth, duckweeds, and water lettuce azolla. We can use them as a remediated plants, as they can grow very rapidly and they can accumulate in high biomass and strongly nutrient uptake. They can uptake the nutrients from the wastewater and make the water clean. So we can utilize the nutrients as well as we can remediate the wastewater also. So the mechanisms by which the aquatic macrophytes are remove the pollutant is the phyto-extractions, where the plant extract the pollutant from the water and accumulate it within the body. And by removing the body, we can remove the waste from the water. And another mechanism is the rhizofiltration, where the roots secrete several type of compounds that helps in the precipitations or absorbance of the pollutants in the water, and they becomes unavailable for the water body or other parts. And another mechanism is the phytodegradations, where the plants secrete some chemicals which degrade the pollutant into less pollute materials. And another one is the phytostabilizations, where the contaminated is immobilized within the soil body and kept remain within the soils and does not cause any harm to the environment. So these are the mechanisms by which the aquatic macrophytes can remove the pollutants from the wastewater. So these are very efficient also. From the several research review, we have found that they can remove about 90% biological oxygen demand and chemical oxygen demand. Also, the total nitrogen, phosphorus, and heavy metals also from the soil system or water systems, and will be helpful for the wastewater treatment. But the major challenge is the biomass generated of the macrophytes plants. Because if we dump it in any place, it will cause several others problem like greenhouse gas emissions, or secondary some pollution leaks. Some others remove some greenhouse gas, and that cause the air pollutions also, and also the area required for their dumping sites. So these are the problem. So we have to utilize it in a proper way of the biomass generated during the wastewater treatment by the macrophytes. And this can be done by several processes like composting. During composting, it can be transformed into a biofertilizer, which can be utilized in the agriculture sector. And by anaerobic digestions, we can also convert it into biogas or digestate, which can be also used for the energy and for the plant nutrient purpose. We can also convert it through the pyrolysis to a biochar or syngas, and that can be used in the agriculture and also the energy sector. And several others like hydrothermal carbonizations, which produced in hydrochar, that required very less amount of that required water but less amount of heat. And these are the several steps we can utilize it in several ways for the energy and for the agriculture purpose as a source of nutrients. So this will create a circular bioeconomy model where the biomass can be utilized and where the wastewater is purified. So this will be win-win situations for the current scenario, as the surroundings are polluted by the several industry and other things. By these processes, we can also environmental benefits like the water can be utilized in the agriculture sector also. We can reduce the greenhouse gas emissions also, and the eutrophication problem, which is a major problem in the water body, that will be improved by these processes. So these are the environmental agricultural benefits we can get from the phyto-remediation by the macrophytes. But there are several challenges also, because these some macrophytes are not decomposing. Some macrophytes are very high containing of the lignin, although they do not decompose easily. So it's a very problematic. So and also we have to manage it in a high biomass. So there is a need of the different government policy, and also the current trend of the different IoT or AI-based system we can utilize to monitor the systems. So by monitoring the systems, we can do the circular bioeconomy model for the wastewater treatments, and that can be utilized for the generations of the biofertilizers and as well as also some energies. Thank you, sir.
Thank you, Dr. Asif. Dr. Monica, any query for Asif sir or some discussion point?
Yeah, I have one query. Thank you, Dr. Ashif, for your nice presentation.
Thank you.
I have one query is that which aquatic macrophytes you used in your study?
I don't do any study, ma'am. It's a review paper. So from the review paper, I am presenting it. It's not any research paper.
Okay, any parameters?
No, ma'am.
you studied?
No parameters has been done, but I am planning a work to produce the to utilize the biomass to produce biofertilizers. So I am planning for this research, but not till now I have done anything about this topic.
Okay. Thank you, Dr. Ashok.
Thank you, ma'am.
Thank you, Asif sir. As per the list available for the participant, we can only see 4 of them are remaining, but only one is available, Divya Gautam. So, ma'am, can we start with Divya?
Yeah, you can start.
Yeah. Divya,
Yes,
can you
ma'am.
continue?
Yes, ma'am. Mera-
Asif sir, can you stop your presentation view? Dr. Asif?
Slide are visible.
For me, it is not convenient. Dr. Monica, is it available for you?
Dr. Viren, Dr. Asif is here.
Yeah, he is here.
Okay, so can you share? Please switch off your.
Yeah, I have already instructed. Yeah. Ashu sir? Can we coordinate from can you coordinate from there for a presentational view switching off? I want that Muhammad Asif sir presentation which is available. Can we switch it off from your point of view? From your point? Okay.
Ashutosh sir, please call to Mohammad Asif sir na, to stop their presenting. Please call.
Yeah.
Yeah.
Okay, sir.
Now you start. Viren sir,
Yeah.
we start with the next presentation of Divya.
Okay. Divya, can you share up your presentation? Ms. Divya, am I audible?
Divya, are you there? Divya Gautam? Sir, please
I
wait
think-
for 1 to 2 minutes.
I think
Technical
her
glitch.
microphone is off here.
Yes.
Yeah. Yeah, now she is available.
Okay.
Divya, can you share up your screen for presentation?
Yes. Yes, sir.
Divya, click on presenting screen icon.
Divya, are you here?
Divya, please share your screen first. I will call her. Just a minute.
Okay, ma'am.
Mr. Prince Chaurasia, are you available here? No, I guess. Mr. Sanjay Kumar?
Yes, Divya is presenting now the screen.
Yeah, ma'am. I'm just confirming that whether the participants, remaining participants are there or not.
Yeah. Please.
Yeah. Gumandi Brijwasi, sir, are you available? No, I guess. So only Divya is remaining. Sorry, ma'am, if she'll connecting within the 2 minutes. Yeah, Ashutosh sir, you're audible. Ma'am, Dr. Ashwini is there only. She is helping her out in presentation because this is the final one. For this session, this is the only remaining. Okay. Okay, sir. Yes, ma'am, it is visible.
Just a minute, sir.
Yes, ma'am, we are waiting.
Good afternoon, sir.
Good
Good
afternoon.
afternoon, sir. Sorry.
No issues.
Thank you, sir. Thank you, ma'am. Good afternoon, everyone. Now I'm going to talk my presentation that I have prepared. The little of my project work. The title of my project work is study of microbial mechanism involved in bioremediation of heavy metal from cement affected agricultural soil. First of all, I want to introduce little about my topic. Heavy metals. Heavy metals are elements their density, high density and toxicity that accumulate in soil and harms to plants and animals. 8 minutes. Heavy metals are elements that are high density and toxicity at the even low concentration. Some examples are lead, cadmium, chromium, mercury, and nickels. And I have some examples. Heavy metals tolerance microbes are Pseudomonas, Bacillus, Rhizobiums, and Aspergillus, and Penicillium. Sources of heavy metals contamination. Main sources include industrial waste, mining activities, cement industries, agriculture, fertilizer and pesticides, sewage, sludges, etc. Then isolation of microbe from soil sample. It's cement affected soil. The sample are collected from Satna, cement factory. Collection of soil samples from cement affected soil. About 1 gram of soil samples are added to 9 ml of distilled water to prepare the initial suspension. Then sample are mixed properly using a vortex or shaking method. Then serial dilution was performed. 0.1 ml sample was sprayed on nutrient agar plate using a sterile spreader. Plates were incubated at 28 to 37 degrees Celsius for 24 to 48 hours. After incubation, different bacterial colonies were observed. Distinct colonies were picked and streed on fresh nutrient agar plates to obtain pure culture. Morphological identification. First, gram staining. Gram staining are a technique that will on the basis of bacteria cell wall. Thick cell wall bacteria, that is gram positive bacteria, and thin is gram negative bacteria. On the basis of color and on the basis of shapes, the gram staining process techniques is done. Biochemical tests are performed: indole test, catalase test, citrate test, and MRBP test. Then indole test to check the ability of bacteria to degrade tryptophan into indole, and catalase test to detect the presence of catalase enzyme in bacteria. Citrate test to determine if the bacteria can use citrate as the soul of carbon soul. MRBP test to detect the type of glucose fermentation performed by bacteria. There are some images of a biochemical test. In the previous slides, there are some biochemical tests. Some is negative and some are positive. After the result, further perform PGPR test, that is a plant growth promoting rhizobacteria. PGPR test. PGPR tests are used to identify bacteria that promote plant growth by producing growth hormones, nutrient and protective compounds. Common PGPR test includes IIA productions, phosphate solubilization test, nitrogen fixation test, ammonia production test, HCN production test, and SIDO4 production test. Then, isolation of microbe from cement affected soil has been completed. Biochemical characterization has been performed. PGPR tests are currently in process and final results are yet to be obtained. Future purpose and future scopes. To identify effective microbes capable of removing heavy metals from cement affected soil, to understand microbial mechanism involved in bioremediation of heavy metals, to develop eco-friendly method for detoxifying cement-affected agricultural soil, to apply plant growth promoting rhizobacteria for sustainable agriculture and environmental protections. Thank you so much, sir and ma'am.
Thank you, Ms. Vivya. You have presented very well. And over to Dr. Monica for Q&A session.
Thank you, Divya, for your presentation. And I have one question here. What is the citrate utilization test, Divya?
मैम सिट्रेट यूटिलाइजेशन टेस्ट में हम सिनेमन सिट्रेट अगार यूज करते हैं मैम, जो कि हमें वो लैब में इजिली मिल जाता है. दैट इज अ ग्रीन कलर, और उसमें जो बैक्टीरिया होता है वो कार्बन यूज करके ब्लू पिगमेंट देता है.
Okay.
Any question, ma'am?
Any test method you have done for the heavy metal removal?
मैम heavy metal, yes ma'am.
कोई दूसरी मेथड जो कि आपने यूज किया है अभी हैवी मेटल रिमूवल के लिए?
No ma'am.
Okay.
ठीक है मैम. Thank you, ma'am.
Thank you, Divya.
Okay.
Thank you so much, sir.
Yeah, thank you. So, as per the list provided for the presentations of today, we have Dr. Pranita, who has started this session, then followed by Anurag Tripathi, then Banti, Shivani, Rashi Mongia ma'am, Dr. Mohammad Asif sir, and Divya. We have 3 more remaining, but they are not available in the session today. So we'll be having the final call from Dr. Ashutosh, Mr. Ashutosh sir, that whether we can stop this session here only or we need to wait for. Okay. Okay, sure, sir. Okay. So with the permission from Mr. Ashutosh, we'll be closing up session here only for the International Conference Track 1. And before winding up the session, I would like to thank all the participants who were available during the session for their patience shown in this particular session of 1.5 hours. Thanks, Dr. Monica Masoni. She has provided complete 2 hours for our session. And the first presenter, Dr. Vishnu Omar, he has presented as well with the first talk. And to all the participants, we'll be continue for track 3 and 4. But from track 1, we are completing the session here only. So thank you, everyone. And the viewers, will be continuing this conference till the validity session. Okay. Thank you, ma'am.
Thank
And
you.
thank you, Mr. Ashutosh. Okay, sir. Okay.